close to our kind of attraction of the evening. It's a great pleasure to have Professor Paul Selvin give a keynote speech for our summer school tonight. And I have here his CV that basically is about 50 pages, so it will take me a few minutes. I'm kidding, I'm going to read just a couple of points here. So, Professor Selvin received a PhD in physics from Berkeley, and the thesis was DNA dynamics from picoseconds to milliseconds. He received a bachelor's also in physics from University of Michigan, Ann Arbor. In terms of research accomplishments, he's a world leader in single molecule imaging, and in particular, his groundbreaking studies were on developing Fiona, which I think a part of it will be discussed in the presentation today. And basically, this allowed 1.5 nanometer spatial localization of single fluorescent molecules with sub-second temporal resolution. He was the first to determine the hand-over-hand versus in-truer model for single molecule myosin 5. That was a famous science paper in 03. Myosin 6, that's another paper in 04. And kinesis, another science paper in 04. Determination of step size and rate of stepping of kinesin, dining in vivo, that's a science paper in 05. I could do this all night. Less than 8 nanometer spatial resolution between two identical fluorescent dyes. This is another 04 Keynote paper. The list goes on and on, but I think I will just stop here. I'd like to just mention that actually Professor Selvin is also a very good ping pong player. We're playing ping pong sometimes. Actually, we had to skip it tonight because of this event. So thank you so much, Paul, for coming, and it's a great pleasure to have you here back.
Thank you again, Gabby. It's fortunate that this happens to be on a Thursday night because we have ping pong on Tuesday and Thursday and Saturday morning. So you shouldn't be too disappointed. My son managed to drive me here so he could take the car to go to ping pong. While I don't think I'll be able to compete with the 6 wonderful videos that were given, I'll try to keep you somewhat entertained. What I'm going to do is I'm going to tell you about single molecules. And in particular, I'm going to tell you about 2 techniques. One is the in vitro fluorescence, which we largely did via what we call Fiona, and I'll explain that in a little bit. And one we have that is really totally new, which is in vivo meaning in the cell. It's an optical trap, where an optical trap is usually only useful for in vitro. And this is the first time that we've managed to successfully apply it in vivo, and we actually got something worthwhile out of it. Okay. I want to right away thank Mindy Hoffman and Ben Lem, who are the 2 graduate students who did the work. Of course, I also like to thank the people who funded us, the NIH and the NSF. And that's Ben. That was a few months ago. And that's Ben now, where he's graduated. It's the first time I've ever seen him dress in a suit and tie. And then we have Mindy, who also graduated Turns out last December, because she had quite a task to do to get out in December. Because in October, she gave birth to a baby girl. Isn't that cute? Okay. So now for a little bit of science. What we're going to be doing is studying molecular motors. Molecular motors are things which take cargo and move them from point A to point B. And in general, diffusion is not a good way to do it. Part of the reason is in eukaryotic cells, which are, let's say, at least 10 microns or so across, it's just too far to do it. It would take essentially forever. Plus, it's difficult to go actually from a specific point where a protein, for example, is born to another place where the protein is actually doing the action. So what evolution has done has invented what are called molecular motors. And what we're going to focus on are molecular motors that operate in the cytoplasm of the cell. These molecular motors, they walk on roadways. The particular type of roadway that we're going to talk about today is microtubules, which are shown in green. And in general, microtubules are ordered diametrically, radially. And it turns out they're polarized as well. So going north and going south is different. And going north, you actually use a different car or molecular motor. If you're going plus towards the cell membrane, you generally do get on kinesin, it's called. And if you want to go towards the nucleus, you walk on with diamine. So for example, when a virus, cold, invades you, it latches onto the outside of the membrane, it gets through the membrane, and then it goes and recruits some diamine, which then bring it to the nucleus. It hops into the nucleus, incorporates itself in your DNA, and there you have it for the next 7 days or so. You have a cold. Okay. One thing that I want to point out is this is often the diagram that's shown of a cell with lots of space in there. But in reality, in fact, the cell is an extremely congested place. It's much more like there, which, by the way, for those of you who are familiar with this, I initially said this was the Golden Gate in San Francisco. And I lived around San Francisco. Turns out that it's not the Golden Gate, it's the Bay Bridge. But if you happen to come from San Francisco, you'll know it doesn't matter what bridge you're going on, the thing is always congested, always looks like this. Okay. So the fact that there's so many roadblocks is actually going to be important. And just to fill you in a little bit of background, you have kinesin, which walks north, or so-called walks in the plus direction of the microtubules. You have diamine, which walks in the minus direction. The way, if you're not familiar with it, you can think that you're going to get a D minus because I'm a really tough taskmaster. D meaning diamine and minus going towards the minus. Not okay. Kinesin, it turns out, that walks on one of these protofilaments. As you see, the microtubule is actually made of 13 protofilaments, 1, 2, 3, etc. Kinesin is very good in that it does a little tightrope walk. It almost always stays on the same microtubule. At least this is in vitro. Diamine also takes steps in the other direction. By the way, I don't know if I mentioned, the steps are 8 nanometers in length. This was initially discovered in optical traps, but I'm going to show you that we've done it another way. Okay, so kinesin walks 8 nanometers this way. Diamine walks usually 8 nanometers, but actually it can be quite variable. 8, 12, 16, even 24, 32 nanometers. Furthermore, diamine has had a little bit too much to drink, and that it kind of, it walks kind of staggering, so it doesn't always walk on the same protofilament. And that, it turns out, will also be important. Okay, just to show you that, don't laugh. Just to show you that we can in fact measure 8 nanometers with fluorescence. And of course, normally, you're limited to how fine you can see with fluorescence or any other visible light technique, to normally to about lambda over 2, where lambda is about 500 nanometers. So the resolution is typically 250 nanometers, but in fact, we'll show that we can get 1.5 nanometers anywhere down to a millisecond up to a second. And of course, this was invented by Fiona. Raise your hand if you don't know who Fiona is. Hey, come on.
I don't know who Fiona is.
There we go. Where are you from?
California.
Not very. Anyways, Fiona, when she isn't starring in the movies, stands for fluorescence imaging with 1 nanometer accuracy. And the idea really is very simple. You take a single fluorophore and you image it with a CCD camera. The image will have a width of about 250 nanometers, but if you get enough photons, you can determine the center of that distribution much better than 250 nanometers. And it turns out you can locate it to 250 nanometers divided by the square root of n, where n is the number of photons. So this particular image is 14,000 photons, let's say 10,000 to make life easy. Square root of 10,000 is 100. 250 nanometers divided by 100 is 2.5 nanometers. Or in fact, it ends up being about 1.25 nanometers. So that's really good. It's sort of just like you see an isolated mountain, and of course, you can tell that where the center is much more accurately than the width. And here, for example, we have some kinesin, which is like this, and we put a large fluorescent sphere on it. We actually can do regular size fluorescence, but in this case, we have quantum dots. And you're going to see that it walks. There is individual kinesins walking along. And what we can do is we can just plot where that position is as a function of time. And you see here it's waiting for an ATP, then it gets an ATP, it takes a step, it gets another ATP, takes another step, etc. We can graph this, and that we get that there's 8.4 nanometers per step. And according to optical traps, they're 8.3 nanometers. So that's pretty good agreement. Okay, so there's no question that we can see very small steps. Okay, now let's go in vivo, inside a cell. And already you're going to see some complication. For example, look at this. This guy is going up and back, up and back, up and back. Cargos frequently display this up and back. Furthermore, usually carry both dynein and kinesin. So how do they know which way to go? Takes a few steps in kinesin direction, and it takes a few steps in dynein, and then a little bit more, and then back, a little bit more. And that's the question that we were very interested in answering. The most straightforward way is some sort of coordinated motion where kinesin is going one way, dynein is going the other way, and they have a hormone or a peptide that kinesin, for example, has the peptide, dynein comes off, and kinesin walks, and then kinesin hands the peptide back, hormone, whatever it is, and then dynein walks. Okay, so that's the coordinated model. Notice that there should be a peptide or hormone or something that the cell has. Despite a tremendous amount of effort, no one has ever seen this. So maybe it's a tug-of-war model. Kinesin is pulling as hard as it can, and dynein is pulling as hard as it can. And sometimes kinesin wins, and then dynein wins. Or maybe a couple of dyneins take over, that a couple will outcompete with the single one. So this is called the tug-of-war. Notice it does not require any sort of hormone or peptide being transferred between the two. And then, in fact, there's a synergistic model. Now, the synergistic model is, synergy just means that two or more objects functioning together to produce a result not independently doable, not independently obtainable. When I was thinking about this, I realized I wasn't quite sure of the meaning of the word synergy. So I got this from Wikipedia. Okay, and what I'm going to tell you is that it's a sort of tug-of-war, but actually it's really a synergistic tug-of-war. Okay, so what are we going to do? What we're going to do is we're going to take a fluorescent sphere. We're going to attach kinesin and dynein, and then look at it. And if it actually is going up and back, that would say it must be a tug-of-war, because we haven't added any hormone or peptide in it. And here it is. Notice it's obvious that it's clearly in the computer seems to be having some trouble. But it's clearly moving in up and down that fashion. In fact, you can see here it goes kinesin, dynein, and then kinesin, and then falls off. Here it goes kinesin, dynein, kinesin, dynein, kinesin, dynein. Okay, so this means there's no question that it is, in vitro anyways, a tug-of-war. We'll talk about in vitro in just a second. Okay? And just to make sure, we did an assay just with the bead with only kinesin and goes in one direction, and with only dynein and it goes the other direction. In fact, occasionally dynein goes backwards, remember, because it's kind of this drunken sailor kind of thing. And we were very careful to make sure that the up and back was in fact due to kinesin and dynein. Okay? is we compared it to some in vivo results. So this is some in vivo, and these are in vitro, and you see it's clearly moving up and back, and it's very similar to this. Two of them are like here, two of them happen to be like this, and then one of them like this. So it's pretty reasonable that the in vitro data seems to, is the word recompensate? I don't know. Corroborate. Thank you. You can have your name on the paper. Okay, now we're going to talk about how it is in fact must be a modified tug-of-war. Back in 2008, Lukowski published in the lousy journal called PNAS. I mean, it's a great journal. What he did is he showed theoretically that a tug-of-war could possibly answer how the cargo moves up and back. But one of the things in his model was, notice when kinesin is going, diamine is just letting go. So kinesin is just going and it's walking with 8 nanometer steps. And similarly, when it's walking in the minus direction, diamine is going and kinesin is just letting go. And what we find is that's not true. In fact, we find that diamine is really tenacious. That is, it tends to stay on the microtubule all the time. Whether it's going this way, or it's going that way, diamine is on the microtubule. And it's different. This is a synergistic effect. It's different than kinesin in that kinesin, it turns out when it's going in the minus direction, so it's being pulled backwards, kinesin has a tendency just to come off. So, how do we, how do we show this? Well, what we do is we're going to apply a force. Basically, we'll take just kinesin, we'll see it's moving in the plus direction, and then we take a force, in this case an optical trap, and tug it backwards. And either kinesin will come off, or it'll walk backwards. So it turns out that kinesin won't walk backwards, your diamine will walk backwards. Okay. And that's the optical trap. So we have kinesin. We all of a sudden, the kinesin is walking in the plus direction. We pull it back, and what we see is you get a sudden release. So kinesin doesn't walk backwards, as was sort of expected. But on the other hand, diamine, actually not just any diamine, but actually mammalian diamine, walks backwards. That is, you're going in the minus direction, and then you give it a tug, and what you actually find is you look this, and you look, and there are a series of big steps. And remember, I mentioned that diamine can take 20 nanometers or 30 nanometers and so forth. So apparently, when you tug on it, make it go backwards, actually, rather than falling off, it stays down. Furthermore, yeast diamine, turns out that there's not many different types of diamine. It's very different, actually, than kinesin. basically there's not many different types of diamine, but yeast-diamine happens to be one of them. Yeast-diamine is basically there to pull on the entire nucleus in yeast. Yeast-diamine does not. Okay, so we have a model where if it's walking in the kinesin direction, the diamine stays attached. If we're walking in the diamine direction, diamine is of course attached, but kinesin comes off. But there must be more, because it can now back up, that is diamine stay up. I have a little computer problem, and apparently some of my cool cartoons are missing to that.
We can change the file.
Don't even talk to me about that file. Okay, what I had. There is an old car that Mindy, who did this work, used to have. Honest to God, it was an old car such that you put in the transmission, you put in drive, and it would go just fine. You stop, put it in reverse, nothing happened. So let me tell you, there were some interesting situations where all of a sudden she had to back up, and out comes the people. So it's clearly advantageous for diamine to always be there to back up in case you have to. Okay. But now, how do you go past a roadblock? Remember I said that the situation in a cell is very crowded. So you're walking a microtubule. Sometimes you're taking something huge, like a micron or 5 micron big, and you're going to naturally run into actin, microtubules, etc. And yet, somehow, it gets by, because it's known that in the cell, the kinesin or diamine can move essentially as fast as the kinesin or diamine in simply water. So, how can you do this? What we did is we made a series of roadblocks. In one case, this roadblock was other microtubules. So there's a microtubule, there's another microtubule. Notice, it's going, and then whoop, whoop. Whoop, whoop. Want me to do that again? Okay, so what's actually happening here? This is a plot of the position versus the time. And what is happening is, this way is north, so kinesin is going, and then it stopped. Diamine takes over, then kinesin takes over, and goes right through. And so that's actually pretty reasonable, because you think kinesin is a straight walker. Remember walking on a single protofilament? All of a sudden, boom, it hits something. So it has to stop. Because diamine is right there on it, the kinesin eventually comes off, and the diamine takes it and moves it back. But because diamine is like this drunken sailor, it happens to go on different protofilaments. So it goes on different protofilament, and then at some point it stops, kinesin takes over, and it goes now on a different protofilament, but the roadblock was over here, not over here. So it can manage to get by a roadblock. So this is what we call the synergistic effect. So that, pretty good. I learned the definition from Wikipedia. Okay, not only microtubules, but in fact, we did a single microtubule. It turns out that there are MAPs, microtubule-associated proteins, all over the place. And in fact, we saw a very similar thing. Notice it goes kinesin, it stops, it goes undynein, and at some point it stops and kinesin takes over and away it goes. So it's important. It's this transfer from the kinesin to a dynein and then back to kinesin, which we would argue allows it to bypass roadblocks.
So when they jump photofilament, when you have roadblock, they jump another like dynein will jump to another photofilament. Will that kinesin that was on the same photofilament, it won't necessarily jump to the same photofilament?
No, it also will not necessarily.
It's just random, right?
That's right. Yeah, presumably just random. That's right. And so, in fact, oftentimes it hits a roadblock. It backs up with dynein. Most of the time, in fact, dynein walks on the same protofilament. And if it does, kinesin then takes over and it's stopped. And then it goes back and it has to hit it many times until it does transfer to a protofilament that allows it to walk by. Okay, so this is the conclusion of the first part of the talk. By the way, don't worry, we're more than halfway through it. How about if you really want? Okay. We can replicate bidirectionally the saltatory, that means the up and back motion, with our simple in vitro system. No regulatory complex is required. Therefore, it supports a tug-of-war model. But reversals allow the cargo to overcome obstacles. It hits it with kinesin, it goes back, occasionally moves across, and then goes. So dynein's ability to walk backwards is likely key to achieving the saltatory type of motion, which we saw early on, and navigating obstacles in the cell. And an interesting fact here, if you kill one motor, let's say you add RNAi, which has the effect of killing just one motor, what happens? This has been done a lot. It turns out that all motion stops. You might think, okay, if I kill dynein, that all motion would stop when the thing has gotten to be outside of the cell, because kinesin would take over. That is not what's happening. Similarly, if you kill kinesin, you might think, okay, all motion would stop, but only after. Is that you?
No.
But, because, I might have to take you off the paper. Okay, and so this has been a very big mystery why this is, but this potentially answers that question, that all motion stops because often there's an obstacle in the way, and so it just stops right there. Okay. Notice the synergy. Okay. Now I'm going to talk about the in vivo trapping, which actually, fortunately, is completely consistent with the in vitro result that I just got. Can you imagine you know, you work so hard to get the in vitro result. Imagine then if you got something that was diametrically opposed. Fortunately, that didn't happen, and we can submit them both to the same journal. And then, of course, what the journal wants, that this honestly happened. They said, why can't you just combine it into one article? It's like, you know, well, there were 2 graduate students working on it, you know, and each of them want to get credit for each of their own thing. It was difficult, but she agreed. Okay, and again, this is just to remind you what inside the cell looks like. The green is the microtubules, the red is the actin, that there's all sorts of roadblocks inside the cell. What we're going to do is we're going to do with an optical trap. Raise your hand if you know what an optical trap is. Okay, mostly, okay, that's good, because actually I meant something.
People in California might have.
Some of my talk went out.
Okay, essentially what you do is you focus down a laser beam to a diffraction limited spot, and things will tend to get pulled into that spot. Essentially it acts like a spring. And in fact, here you have a motor walking this way, the spring is due to the light, and you can see, turns out, very small steps, like, for example, they saw the 8-nanometer steps. You can also wait until it walks, because the force equals k times x. Once x gets big enough, the force gets large enough that the motor can't walk. And that's called the stall force. At some point, it's as likely to go forward as it is to go backwards. And it turns out for kinesin, the stall force is on the order of 6 or 7 piconewtons. For diamine, actually, it's more of a controversy, but it's probably just 1 piconewton. Okay.
Well, he's drunk.
I'll have to remember that. Okay, so what we're going to do is do just the simplest system, which is in vitro, which has been done before. And we do an intermediate system where, in fact, we'll take the cartridge just like before, and we'll attach kinesin and diamine to it and see what it looks like. And then, in fact, we'll do the most complex, we'll do it inside of a cell. And why is that so complex? Well, it turns out that in water, because it's water, there's a viscous effect. There's no elastic effect. Elastic, like, is, with going through honey, that you stop and then you'll go a little bit more. In water, there's none of that. It's all viscous. In a cell, however, there's a serious issue of elasticity. Elasticity comes from you're walking along in a cell and you hit a bunch of filaments. They can bend, but they'll eventually make you stop. Okay, and so you have to deal with what's called viscoelasticity, and this has prevented basically, I think when the original one was done was 67. It's been like 40 years that people haven't been able to do it, but we have solved it. And I want to say, this is one of the cases where it's a totally royal we. I mean, professors don't usually admit this. In fact, what I should say is when I say we, I really mean he, meaning Ben Glenn. He's done it all. I feel kind of bad. No, it's not the reason that you're thinking of. I feel kind of bad because he came to me and he said, I really want to graduate. And I was saying, well, you haven't quite nailed it down yet. He said, well, Paul, I haven't been here in 9 years. At which point I said, it's time to graduate. Fortunately, he's doing a postdoc. Okay, so what's the answer? The answer is right here. We take a cell, and you take only those that are going north, or the plus direction, going outward. And you see, and you look at what the stall force histogram is. And you actually see some very broad distribution. I'll get to this in a second. You look in the minus direction when the guys are going towards the cell nucleus. And here again, you see actually a broad distribution, though it's a little bit more peaked. So what do these histograms mean? The simplest case, we took kinesin and we did it in vitro, and you see it comes basically pretty narrow, and it peaks at about 6 piconewtons or so. So it's significantly narrower than this, and it's peaked about here, 6 piconewtons. And in fact, diamine over here is similarly peaked, or even narrower, about 1 piconewton. So in fact, it's about over here. So definitely not just your water-based environment. However, what we do is we take a bead and we attach diamine and kinesin to it in vitro, in water. And what do you see? You see something that looks like this, which is just like our cell results. So what this says is this distribution is coming from the fact that you have kinesin and diamine, that is, they're synergistic. It's not due to, for example, that kinesin has, in the cell, has a weird distribution that's really broad. I mean, it could have been, but actually it wasn't. Okay? So in fact, what we think, this is, by the way, in the plus direction, the kinesin stall force is equal to the kinesin alone, that's the 6 piconewtons, minus the amount of diamine stall force. So if you have diamine as a stall force of 1 piconewton, so you have 6 minus 1, and you have 5 piconewtons. And we get some 5 piconewtons. And then diamine, and then you have another situation where you have kinesin minus 2 diamines. So that's 4 piconeedles. Then you have kinesin minus 3 dyneeds, and etc. So you actually get an entire range over here. And that's fundamentally the explanation. This is in the plus direction. Then in the minus direction, you see here's in the minus direction. It clearly is not due to just a single dynene, but in fact it's due to, we think, maybe 2 or 3 or 4 dynenes. With no, you don't have to do any subtraction, because remember we said when the thing is going in the minus direction, kinesin comes off. Where on the other hand, when we're going in the kinesin direction, dynene is staying there, and so if there's 1 or 2 or 3, those stall forces have to be subtracted off. And in fact, so what we did is we took a bead and we added multiple dynenes, and we somewhat got larger stall forces, though in truth it's not perfect. Okay. And so it's really a shame. Okay. Hold on a minute. This is probably going to get me. The that's exactly what I thought. Hold on. Okay. Everybody, close your eyes. Close your eyes. I'm serious. This could ruin the joke. Great, you can open it. Okay. Hopefully this will work. The bottom line, moral of the story, is when you're faced with a roadblock, just don't call a friend. Have a friend with you, because a good friend will come bail you out of jail, but a true friend will be sitting next to you saying, can we? Okay. And again, the people who did the work really, Indy really did a wonderful piece of work. Ben, and they also had assistance with proteins that from Amit Yildiz, actually it turns out he's a former student of mine. He's at UC Berkeley now. Kathy Travis and Chris Berger, who's at the University of Vermont, and Tina Schroeder, who's at Johns Hopkins University. And thank you for your attention.
Thank you very much, Paul. I think we have time for questions. Please.
So, I don't know anything about biology, so I'm going to ask out of ignorance. You said that this proteins, they move because of stimuli from ATP.
Yes.
So that's why you have these steps. So is this an indirect way of tracking kind of the electrochemical transport along the protofilaments, correct?
It depends on the definition of indirect, but yes.
So I was thinking just like to extrapolate the same kind of mechanism, these studies, observations, but in axons and neurons, we have those same kind of structures of microtubules and photofilaments, and they have, remember, Roger Penrose explained to me these observations, why the high speeds of transport of electrochemical signals because of the internal spiral structure of the filaments within the microtubules. So you can probably study in the same way these kind of transport neurons, or no?
Well, yeah, this was, we actually studied two different cells, but they were not as interesting as neurons. And in fact, neurons is something which we're very much interested in getting at and seeing whether the protofilaments, whether they're in fact straight or bent, and what the carbon is doing with them.
Great. Other questions? Please.
I think your explanation of why dining is drunk made a lot of sense. Why isn't it always drunk? Are some of the roadblocks transient? Why does it just back up straight sometimes
Well,
versus randomly
well,
all the time?
what you do is you look at the, first it's a sort of heuristic thing. Let's say, for example, let's say you're looking at kinesin. You know, kinesin is very much a straight walker, and it's always, I say it's always going in the plus direction. In general, that's true. Is it really always going in the plus direction? No. Actually, it turns out about 95% it's going in the plus direction. Occasionally, it does take a step back. And similarly, with dining, although dining is not as processive in that dining more like 80% of the time takes a step in the minus direction and 20% back. In terms of knowing the data just isn't here, but knowing what percentage of the time it takes a straight step versus a wobbly step, it is not actually known, though we hope to do it, though it's a difficult term.
Are the roadblocks sometimes transient?
Oh, yes, actually, I'm sorry, that's a very good point. It turns out these microtubule associated proteins, which are ubiquitous, it turns out that some of them are transient, they come on and off, and others are, they bind and they're there. So in fact, that's another thing that we're interested in looking at, is looking at the map 3, which is this transient one, versus map 4, which is this permanent one, and see how it varies. Yes. And by the way, if anybody is interested, besides Steve, in clinical things, it turns out that a mutation in one or the other affects the neural behavior of things.
The questions?
I got a very simple question. I was surprised actually by the tug-of-war concept because it doesn't look so effective actually. So my question is, what's the rationale for that? So why is that?
Right. It's interesting. I mean, if you think about, you want to go from A to B. I mean, that's the whole evolutionary reason that molecular motives are there. To go, you know, in A and then you go, you know, like that is a very inefficient process. It's very inefficient in terms of using up ATP. But apparently, using up ATP is not that big of a deal. Turns out that in the cell, there's something like 2 to 5 millimolar ATP, which is actually a huge amount, completely saturated. So apparently, the cell is willing to do that to get around these problems of roadblocks.
And is it general among all species?
You know, we haven't tested all species. And when I asked the NIH to do one species a year, at some point they'll stop. I don't know.
Small question, when you have one protein going in one direction, you said that, so I think it was, you said dining is going in one direction, kind of seeing will stay, it's going the other direction, it'll come off. So is this non-local interaction, is known why it's this way, or how does that work? Or is the communication like a longer term, or what is this?
No, the communication happens, we think it's just through the cargo. That's the common theme. It's unlikely to happen through the microtubules. And the cargo is there, and it's tying them together.
Well, there are no more questions. We'd like to thank you for all This was great. So I think we're going to leave the Best Movie Award for the last together. Okay. Okay. And so may I invite some of the organizers to shake hands with our participants who are graduating now. Would that be okay? So I'm reading name by name. If you guys can come this way to the positive side. Pen is inside. That's graduation. Okay. Is that okay? Steve, are you ready? Umberto, Naya? All last time, okay. Donnie Aker. Joe Bailey. Sergio Baggett. Cedric Flatter. Waiter. Cesare Gospania. Oh, yeah. Pavel Ginsberg. Samson Lu. Brendan Wang. Shahidul Islam. Navin Kadasa. Maureen Keenan. Firuzeh Babaye Kurastan. Sorry if I And latrine. Beima. Selomanoto. William Muria. Amir Nahas. Fatemek Nushaban. Jung-Hoon Park. Tessa Pina. Eva Poch. Alessa Second Jan. Amardeep Singh. Jovan. Srinokul Upadhyayula. Nicholas West.